Most things are soluble in water, and blood is mostly water, so it can transport most nutrients, hormones, enzymes. It also removes wastes. It protects against infection. We'll talk more about that when we get to the immune system. It contains buffers, so it stabilizes the acid-base balance, and it regulates the temperature by either sending more blood to the skin. That will make you lose heat to the outside, so that will cool you off. Or it can send less blood to the skin, and that way you lose less heat to the environment and you stay warm. So if we look at the components of blood, you have if you put it in a centrifuge, the heavy stuff goes to the bottom. So at the bottom you'll have the red blood cells, which make up most of the formed elements. Then you'll have a buffy coat, which contains the white blood cells. And then about 55% of it is the plasma, which is mostly water. And then it does contain some proteins. So blood cell formation is called hemopoiesis, and new blood cells are formed in the red bone marrow. All types of blood cells are produced there, and in the lymphatic tissue. We talked about the thymus last time, how it's part of the endocrine and immune system. Well, it helps with blood cell formation too. Blood cells produced in the bone marrow go there to be further developed. Alright, next is the function of red blood cells. They deliver oxygen to cells and they remove carbon dioxide. And if you look at the shape of it, it's called a biconcave disc. And as they're formed, basically they form a nucleus, they make lots of ribosomes, they make lots of hemoglobin, and then in the last step they spit out their nucleus, and that's why they have this shape. So they're basically just a sack full of hemoglobin, which is why they don't live very long, which we'll talk about next. But because of their shape, every point in this cell is very close to the plasma membrane, so oxygen can diffuse in there really quick, carbon dioxide can diffuse in and out. So that's why they have their shape. Next is the structure and function of hemoglobin. So the structure is made up of 4 protein chains, and each one has a heme group. That's where the oxygen attaches. Its function is carrying oxygen. Each hemoglobin can carry 4 oxygen molecules, and there's about 250 million hemoglobin molecules in a red blood cell. So each red blood cell can carry approximately 1 billion oxygen molecules, and you have millions of red blood cells, so your capacity for carrying oxygen is just astronomical. And the amount of oxygen the blood can carry depends on the quantity of red blood cells and also hemoglobin, and those are pretty easy to test. Then we need to know we need an adequate dietary supply of which mineral for hemoglobin synthesis, and that is iron. Then we're going to watch this animation on the life cycle of a red blood cell.
Red blood cells circulate for about 120 days. At that point, they die, break up, and are consumed by phagocytic cells in the spleen and the liver. Every second, about 2.5 million red blood cells are destroyed. Consequently, the body must constantly produce new red blood cells, or erythrocytes, to maintain homeostasis. The process of producing new erythrocytes, called erythropoiesis, is maintained through a negative feedback loop. The process begins when oxygen levels decline, as damaged red blood cells are removed from circulation. The kidneys detect the declining levels of oxygen and respond by secreting a hormone called erythropoietin, or EPO. EPO stimulates the red bone marrow to begin the process of creating new erythrocytes. An immature form of an erythrocyte, called a reticulocyte, is released into circulation. After 1 to 2 days, the reticulocyte becomes a mature erythrocyte. As the number of red blood cells increases, oxygen levels rise. Less EPO is produced, and red blood cell production declines.
Okay, so to summarize that, when the oxygen levels decrease, the kidneys secrete erythropoietin or EPO, which you may have heard of this. Some endurance athletes will use this for blood doping. It causes more red blood cells to be made, and that increases your oxygen carrying capacity, and that's a benefit if you're in an endurance sport. Then the third step, that EPO is going to go to the bone marrow and stimulate it to create erythrocytes. Erythrocytes are the fancy words for red blood cells. Then step 4, reticulocytes are released. Those are just immature red blood cells. They'll mature into red blood cells in 2 to 3 days. Then the oxygen levels increase, and then the sixth step, EPO and red blood cell production are going to drop because we're back in homeostasis. Next, let's watch the breakdown of red blood cells.
As a red blood cell ages, its membrane weakens, becoming fragile. As it passes through the narrow capillaries in the spleen, it begins to break down. Macrophages in the liver and spleen ingest and destroy old red blood cells. As they do, hemoglobin is broken down into its two components of globin and heme. Globin is further broken down into amino acids. The amino acids are used for energy or to create new proteins. Heme is broken down into iron and bilirubin. Iron is transported to the bone marrow, where it is used to create new hemoglobin. Bilirubin is excreted into the intestines as part of bile.
So we're constantly, since they don't have a nucleus, red blood cells don't live very long, so we're constantly breaking them down. The hemoglobin gets broken down into globin and heme. The globin is broken down into amino acids, which are then recycled. The iron travels back to the bone marrow, where it's used to produce new hemoglobin. The bilirubin is secreted into the bile, which actually helps us digest fats. That leads us to this question about jaundice. Jaundice is when excess bilirubin enters the tissues, causing the skin and the sclera, that's the white part of the eye, to look yellowish. This can be from liver disease or bile duct obstruction. It's also common in newborns, so when you're a fetus inside the mother takes care of getting rid of your waste for you and getting rid of bilirubin for you. Once that cord is cut, you're on your own, and a lot of newborn livers aren't ready yet, so they use what are called Billy lights. It's actually a UV light which helps break down the bilirubin, and they have Billy blankets, and it makes your baby look like a glowworm, which is kind of cool. So that's jaundice. Next disorder is polycythemia. This is too many red blood cells. It can develop from cancer. It could be from conditions that cause the amount of oxygen in the blood to drop, such as smoking or living in a heavily polluted environment or living at a high altitude. Symptoms of this include headache, ruddiness. Ruddiness is basically you're red in the face, and itchiness. If it goes on for a long time, it leads to high blood pressure, blood clots, even heart failure, because it makes your blood thicker and harder to pump. This is why, for example, mountain climbers, when they're going to climb a super high mountain, they have to go and they have to get acclimated to the altitude for a couple weeks. That gives your body a chance to produce more red blood cells, since the air is thinner up there. Next disorder is anemia, which is not enough red blood cells. Could be caused by blood loss or by nutritional deficiencies. So without enough red blood cells or hemoglobin, the oxygen carrying capacity of the blood is diminished. This causes fatigue, pallor, which is very pale skin, and possibly shortness of breath. The blood viscosity or thickness is reduced, resulting in a faster heart rate and lower blood pressure, and that can lead to things like fainting. Alright, next we need to know the function of white blood cells. They're also called leukocytes, and they protect the body against pathogens. We'll talk all about that in the immune system. Cancer of the blood or bone characterized by an extremely high white blood cell count is known as leukemia. Next we need to know the function of platelets. So they're made from these big cells called megakaryocytes, and they play a role in stopping bleeding. So normally we talk about blood clotting, but before we form a blood clot, we have to stop the bleeding, and that's what we're going to talk about next. Let's go to the animation.
When a blood vessel is cut, the body must react quickly to stop the flow of blood. Immediately after an injury, smooth muscle fibers in the wall of the blood vessel spasm. This constricts the blood vessel and slows the flow of blood. Although the response is only temporary, it gives other hemostatic mechanisms time to activate. The break in the blood vessel exposes collagen fibers, creating a rough spot on the vessel's normally slick interior. This rough spot triggers changes in passing platelets, transforming them into sticky platelets. The sticky platelets accumulate at the site of the injury, forming a platelet plug. The platelets also secrete several chemicals. Some cause the vessel to constrict further, whereas others attract even more platelets. The platelet plug forms a temporary seal in the vessel wall. A more stable solution requires the formation of a clot. Blood clotting, or coagulation, involves a complex series of chemical reactions using proteins called clotting factors. The exact sequence of events used to form a clot varies, depending on whether the clotting process has been stimulated by factors inside or outside the blood. When areas outside or extrinsic to the blood, such as the damaged blood vessel or surrounding tissues, release clotting factors, a cascade of events called the extrinsic pathway begins. When factors within the blood itself, such as the platelets as they adhere to the collagen in the damaged vessel wall, activate the clotting factors, a cascade of events called the intrinsic pathway begins. The extrinsic pathway activates blood clotting factor X in single reaction. The intrinsic pathway requires 4 different reactions to activate factor X. Either way, once factor X is activated, the formation of a blood clot follows a common pathway. After factor X is activated, an enzyme called prothrombin activator is produced. Prothrombin activator acts on a globulin called prothrombin factor 2, converting it to the enzyme thrombin. Thrombin transforms the soluble plasma protein fibrinogen into fine threads of insoluble fibrin. The sticky fibrin threads form a web at the site of the injury, creating a clot of fibrin, blood cells, and platelets. A blood clot can effectively seal breaks in a smaller vessel. However, blood clotting alone may not stop a hemorrhage from a large blood vessel.
Okay, so the first step, hemostasis, is stopping the blood bleeding, and that starts with the vascular spasm. The smooth muscles around the blood vessel constrict. That slows down the blood, and then there's less blood to bleed out. Then the platelet plug is formed, and once one platelet sticks, it releases chemicals that attract more platelets, and then they release chemicals that attract more platelets, and you get this positive feedback loop that keeps going until we have this platelet plug, and that's a temporary stop to the bleeding. And then we need the blood clot, or coagulation, and you just need to know the last steps of this. So you have in the blood, in the last step of coagulation, thrombin transforms the soluble plasma protein fibrinogen into fine threads of insoluble fibrin. And then fibrin, so fibrinogen is soluble, it's dissolved in the blood. Fibrin is insoluble, so it forms this web, and it traps red blood cells in there, which is why clots or scabs look red, and that permanently stops the bleeding. And the liver needs which vitamin in order to synthesize clotting factors? And the answer is vitamin K. And the first step in hemostasis is vascular spasm. Next, we're going to talk about blood types. And let's just go to the animation.
The surface of red blood cells typically carries a protein called an antigen or agglutinogen. There are 2 types of antigens, known by the letters A and B. People with type A blood have the A antigen
blood types are determined by the type of antigen that the cell has. So if you have A antigens, you're type A. This figure shows the antigens and antibodies. So a person with type A blood has A antigens and anti-B antibodies. Then a person with type B blood has B antigens and anti-A antibodies. So the antibodies are kind of the opposite of the antigens. Then a person with type AB blood has both A and B antigens, and they have no antibodies. And then a person with type O blood has no antigens and both anti-A and anti-B antibodies. And the reason it's harmful to give someone with A blood a transfusion with B blood, we can see here if you introduce the B antigen, the B antibodies are going to bind to it and cause agglutination. That can block blood vessels. It will also cause the blood vessels to break and release their hemoglobin, and that can lead to kidney failure, and it's not good. Alright, type O blood is considered the universal donor because it contains no antibodies. Type O blood is the universal donor. I'm looking at the wrong picture. Type O blood is the universal donor because it contains no antigens. So that means whatever blood type, you can receive it because there's no antigens for your antibodies to react with. Then type AB blood is considered the universal recipient because it contains no antibodies. So it could receive blood from anyone because it doesn't have any antibodies to attack that blood. Alright, next thing is RH. So let's watch the animation on this.
Besides being classified according to ABO type, blood is also classified as being Rh positive or Rh negative. Rh positive blood contains the Rh antigen. Rh negative blood lacks this specific antigen. Blood does not normally contain anti-Rh antibodies. However, it is possible for someone with Rh negative blood to develop anti-Rh antibodies. There are two ways this can occur. The first way is when someone with Rh negative blood receives a transfusion of Rh positive blood. The recipient's body interprets the Rh antigen as something foreign and develops antibodies against the Rh antigen to protect itself. If the recipient has a subsequent infusion of Rh positive blood, the anti-Rh antibodies that form during the first transfusion attack the Rh antigen in the donor blood, causing agglutination. The second way someone can develop anti-Rh antibodies is when an Rh negative mother becomes pregnant with an Rh positive fetus. Because maternal and fetal blood does not mix, the first pregnancy with an Rh positive fetus will proceed normally. During delivery or miscarriage, the fetus's blood often mixes with that of the mother, thus introducing Rh antigens into the mother's bloodstream. The mother's body responds by forming anti-Rh antibodies against this foreign substance. If the mother later becomes pregnant with another Rh positive baby, the anti-Rh antibodies can pass through the placenta even if red blood cells cannot. When they do, they will attack the fetal red blood cells, causing agglutination and hemolysis. This will produce severe anemia in the infant, called erythroblastosis vitalis.
Alright, so the conditions leading to Rh incompatibility between a mother and a fetus. So 85% of people are Rh positive. So if a Rh negative woman marries an Rh positive man, it's possible their child could be Rh positive. The first baby's fine, but during delivery the blood will mix, and then that mother will produce antibodies to Rh. So then if she becomes pregnant again, her Rh antibodies can attack this baby, and that usually doesn't happen until birth, but then this baby's born with basically all its blood cells destroyed, and this baby would need a complete